Hi, Honors Bio students. This is Miss Caylor, and this is a short video on how scientists seek truth and avoid bias. And to start, I wanted to just share this image that we're using in class, just as a reminder that not only do scientists try to avoid bias in their own experimental design as they acquire new information, but we all need to carefully avoid bias in how we obtain our information from outside sources. And I thought this was kind of interesting. Speaking of outside sources, I found this infographic a couple of weeks ago. It's a little bit of a sidebar, but it's connected. There was a New York Times opinion piece where they claimed to be able to predict your political party affiliation in America based on a series of pieces of information about you, particularly affinity groups or certain identifiers like race and religion and what part of the country you grew up. And this was based on census data, and there does certainly appear to be a pattern, but what I took away from it was more how our tendency to spend time with people that we maybe share some of these identifiers with can actually be one of the reasons why our biases tend to be very persistent. So if you tend to spend all of your time with people who share your race and your gender and your religion, you're going to just be sharing a lot of the same ideas. You agree with each other. And sometimes that can be really comforting, but that can also be kind of dangerous because you won't be exposed to other ideas. And this is one of the places where bias can really be insidious and persistent. So I want you guys to really be aware of that. And instead of taking comfort in spending time with people who echo your own opinions, think about being open to other opinions. All right, so to talk about how scientists avoid bias, I want to start with this fun story. So you've probably heard of animal magnetism before, and you've probably also heard the word mesmerized before. And it turns out they're actually associated with the same story. So you may think of mesmerized as being hypnotized or in a trance, and that's really what it's all about. So this guy on the left in this picture is Franz Friedrich Anton Mesmer. He was German, and he lived in France in the late 1700s. And he claimed to be able to cure people of their illnesses based on an idea called magnetism. And now he believed that there was this magnetic field, this magnetic force throughout the world. And he was right about that, but he claimed that it was in our bodies and that misalignment or blockage could cause us to be ill. And he believed that through a series of magnet wavings, baptisms in pools with magnetic filings, that he could cure people. And it actually took hold. A lot of people really bought into this nonsense, which we now realize it was. And you can understand maybe why. At the time, medical science was not very advanced. And so many of these people, their alternative would have been to go to a physician and be covered with leeches. So it doesn't sound great, and maybe just dipping your feet in a pool with some magnetic rock sounds a lot more appealing. But they really bought into it, and so it was decided to have a commission to investigate the validity of his claims. And two of the scientists involved in that investigation were none other than Benjamin Franklin and Anton Lavoisier, who you probably all know of as the man who's credited as being the father of modern chemistry. Well, they designed a really cool experiment. They figured that it's possible that Mesmer was correct and that, or he was telling the truth, and that there was some physical thing, this magnetism, causing this curing. People would go into this curative crisis, which was like a trance or almost like seizures, and claim that they felt better. So maybe it was actually magnetism. Or maybe it was psychological. So they said it was either imagination, psychological, or it was magnetism. So what they did was they tested people. They did a variety of tests. They would actually use magnetic solutions or objects that had been exposed to magnetism. And they would tell the people that they were treating them with magnetism. Or they would treat them with something that was not at all magnetized. And they would tell them that they hadn't been exposed to magnetism. But then they also treated people with magnetic objects and told them it was not magnetic, and vice versa. And that was the exciting one. The ones where they treated them with objects that just weren't magnetic at all, but told the people they were giving them the magnetism treatment. And those people, as you might imagine, went into these curative crisis fits and claimed they felt better. And so what Lavoisier and Franklin figured out was basically the placebo effect. And they also essentially did the first blind experiment. So what's a blind experiment? Well, the subject didn't accurately know their role. So those who thought they were being magnetized had something called a placebo effect. They thought they should feel better, and so they did. The ones that were treated with actual magnetism, but were told it was nothing, claimed nothing was happening. So they were able to establish that the magnetism wasn't doing anything. So if the subject doesn't know their role in an experiment or study, they are unable to influence or bias the results through their own actions or reporting. And so we'll see something similar to that when we talk about control groups, a way of preventing the subject, the thing that we're studying, from influencing the outcome. And there's an even better way to do it, and that's the double blind. When we do a double blind experimental design, the researcher themselves is in the blind. And so what that means is that when they collect the data, they are unaware of how the individual samples correspond with the independent variable. So they don't know which subject is receiving the placebo or which subject is receiving treatment A versus treatment B. They'll usually use some coding, then some number-letter combination, and only after they've collected the data and analyzed the data will they then open the sealed envelope and expose what is really happening, and then they can draw their conclusions free of bias. Something else I want you guys to think about is how to avoid misleading graphs. We're going to talk about how we can communicate our findings in a clear way that doesn't that isn't influenced by bias. So this is just an example. If you look at the graph on the left, it certainly looks to be a bigger difference between group A, B, and C, you guys look right here, than the group on the right. But in fact, these graphs are communicating exactly the same thing. Now, sometimes it may be appropriate to do this, and this is called truncating the graph, but sometimes it's not. And you can see that by truncating it, you made this difference, the author of this graph made the difference between 62 and 54 look a lot bigger. So what we're going to try to do then is be really transparent, and we're going to be really thoughtful about how we communicate that information. So we can just present the means like they did in the previous graph. So this is just either a single data point or it's a mean. But we can avoid the problem of putting in different scales on axes by just presenting one more thing. And that's what's the degree of uncertainty. So the mean is what's typical, and you could also use a median. But the degree of certainty is how variable your data was. So you can make something called a box and whisker plot, which is your homework tonight, or you could plot mean and standard deviation, or mean and 95% confidence intervals, and I'm about to show you mean plus standard error for error bars. You can make those in Excel or Numbers or Google Sheets, but I definitely want you guys to all be using Excel this year. So this is an example. This graph is showing average global surface temperatures, and we can see the black line here is recorded averages. Well, this gray area, just kind of upon this gray area, is communicating our uncertainty around those values. So back in the early 1900s, our instruments may not have been as precise, or we may not have had as many data points all over the world as we do currently. So in the latter half of the 20th century, we have really very precise data. So this is a really reliable trend line. You can see it's pretty tight out here, but everything out in the color range is actually a prediction based on different assumptions. So this bottom one is the assumption that we don't continue to add CO2 to the atmosphere, that we level it off. And so the solid line is the prediction, but the lighter yellow is a recognition that there's uncertainty. And that uncertainty, if you guys look at the blue and the green, tends to get much bigger the farther we're extrapolating from our actual data. So I don't want to get into climate change with you guys right now, but I do want to talk about the importance of transparency. So this is the average based on our data, but we can see that our certainty around that value gets wider and wider, our uncertainty, I should say. So I made these two graphs. This is fake data, but I thought it would be a fun way to illustrate. Let's pretend there's a pill that a pharmaceutical claims will make you smarter. So let's say we take a group of male students before, and we measure their IQ. And IQ is not a reliable way to measure, but it used to be used, and it's an easy metric. So we're just going to use it as a hypothetical. So they're measured before, they're given this pill, and then a couple weeks later, they measure their IQ again. And then we do the same thing with a female group of students. If you were to just glance at this graph, and the average American would certainly interpret it this way, you might think that there's a big difference between the males before and after treatment. It appeared to go from an average of 110 to 120 in the male group. But the females also seem to increase in IQ, but they seem to increase in IQ to a lesser degree. But you should probably be asking some questions right now, like what was the sample size? And maybe you're asking, did they only measure people who started at 110, or was there uncertainty around this 110 to begin with? Those would be good questions to ask, because sure enough, in this data set here, the average IQ was 110, but it went up much higher than that, and it went much lower than that in that group. And so what happened here was there appears to be a big difference, but the difference between before and after means is actually smaller than the difference within the group itself. So there's so much variance within the group that you can't conclude that the difference in means is from the treatment. But over here, there's a tight cluster around 110 versus 115. And so you can make that conclusion that they were so close to 110 and they're so close to 115 before, that regardless of the sample size, and sample size is included in this particular measure of error bars, this is called the standard error, this you can conclude. The difference within is smaller than the difference between. And so this would indicate that the treatment had an effect on the female students. It wasn't a big one, but it seems real, it seems legitimate. This appeared bigger, but it's not due to the treatment, or at least we can't tease that out. So you're going to learn tonight about how to make a box and whisker plot. But basically, you need a measure of central tendency, and you need a measure of your uncertainty or variation. We're going to do it with median and quartiles. So you're going to figure out your median, and you're going to figure out your quartiles and plot them both. And then you want to look at the overlap. Significant difference would be one where there's little to no overlap. So when I talk about overlap, I mean to what degree the box and whisker of this plot overlaps with another plot. Kind of like in the previous example where we had the pre-treatment and the post. So in this box plot, we can see that this is the scale bar. We plot the median of the data set. We plot the lowest value, the highest value, and that makes the whiskers. And the box is the lower quartile and the upper quartile. Sorry, that's a little blurry. So if we look at the original data set, this was the males before and after, you can see there's a lot of variation here. So even though the mean was 110, we had individuals as low as 60 and up to 160. That's a lot of spread, and so you can see right there, the standard deviation is 35, and the standard error is 11.6. You don't need to worry about that right now, but it's the standard deviation divided by the square root of the sample size, which, by the way, is only 9, so that's not ideal. I've sorted it, and it's really easy to sort data in ascending order in Google Sheets and Excel and numbers. But here we can see it goes from 60 to 160. So the median, the middle value is 110. The lowest is 60. The highest is 160. And then the lower quartile is 85, and the upper quartile will be 140. So halfway between the median and the maximum, or the number that is halfway between, not halfway between an actual value. So if I plot it, and I used a online option, but you guys have to do this by hand for practice, and I made a mistake, you'll see, but the median is 110, and the lower value is 60, highest is 160, and the lower quartile should be 85, and the upper quartile should be 140.